Hey, Mr. P here.
What's up, guys? It's Mr. Schmitz.
In this video, we're talking about the cell cycle. So what happens to the cell during the cell cycle and how does it go about doing all of its normal biological functions?
Let's dive in. So the cell cycle, by definition, would be the series of stages that results in the production of two identical daughter cells. So with it being a cycle, think of the normal steps that any cell is going to go through as it grows, as it changes, as it prepares to divide, and as it then inevitably divides into two identical daughter cells. So as you can see in this diagram, there are two main phases of the cell cycle: interphase, which makes up most of this pie chart, and then the mitotic or M phase, which makes up a small portion.
And interphase is this kind of teal or blue-green color, but interphase, like you said, consists of most of the cell cycle, and there are subphases within interphase which consist of G1, S, and G2. So when you think of interphase or hear interphase, it is the three main phases that the cell kind of does its normal life in. And so G1 stands for gap 1. That's where the majority of the cell growth takes place. So when a cell divides into two, the two new daughter cells are the smallest the cell will ever be. And immediately upon dividing, each new daughter cell enters G1, its own G1. And so in G1, the cell is going to initially grow and actually increase in physical size. And so if it starts the cell cycle at this size, it will end G1 much larger than it started.
So I would think of G1, for those of you trying to figure out how to remember this, I would think of G1 as like normal cellular function, because the other things that are going to happen in G1 are things like we're going to produce some organelles. So obviously when you come right out of mitosis and you're this little small cell, like Mr. Piper said, you probably don't have as many of the organelles that you might need to really fully function. As you're growing and increasing in size, that requires additional need for more organelles, as well as the production of proteins. So G1 is really what we think of as like normal cell function. If I asked you in class to picture a cell, you'd be picturing it in your mind in G1.
Yeah, skin cells are going to behave like skin cells, and they're going to act like skin cells within this G1 phase. Liver cells, brain cells, nerve cells, any body cell is going to be functioning the way that it's supposed to be functioning in G1. And we've said organelles, but remember back to our cell unit, organelles is kind of a term that refers to a lot of different things. You have the nucleus and the ER and the Golgi and the ribosomes and stuff like that. Moving from G1 into S phase, S stands for synthesis phase, which really the only thing that occurs in this phase. I know it takes up a pretty large portion of the cell cycle, but the only thing that is happening is DNA is being replicated. And so for a cell, this is a vital process in which the DNA molecule has to actually duplicate itself, and it needs to produce two identical strands of that DNA, and so that it can package one of them into one cell and the other into the other cell. One cell with one DNA molecule has to become two cells with a DNA molecule, and so we have to divide the DNA.
And that happens in S phase. After we have duplicated the DNA or replicated the DNA, we move into what's called G2 or gap 2. Gap 2 does some additional cell growth, so the cell will continue to increase in size at this point. We will continue to carry out our normal functions, but we will also begin some prep for division. And so we will produce the proteins, specifically some enzymes that are needed for mitosis itself and the process to divide the cell. So we're beginning to approach cell division at this point in time.
And then once we exit interphase, exit G2, we hit this kind of yellow phase, that's the M phase or the mitotic phase, referred to as mitosis. And so this would be, by definition, the division of the nucleus. We are not dividing the cell yet. We are simply dividing the nucleus and preparing for the end of mitosis, in which the cell does actually divide the cytoplasm. But the mitotic phase is going to include PMAT, or Prophase, Metaphase, Anaphase, Telophase. In the next video, we will go through kind of the intricate details of each of those phases, but for now, this is a very small sliver of the cell cycle in which the nucleus divides.
And like Mr. Pfeiffer alluded to, the very, very last step of the cell cycle is the division of the cytoplasm, which is referred to as cytokinesis. So once we have two nuclei within the same cell, we can pinch the cell in half or split the cell in half depending on the cell type with cytokinesis in order to form those two identical daughter cells, each of which will then begin going through their own cell cycle.
And we refer to these as new daughter cells. The daughter cells, like we talked about in the cell cycle, are identical through mitosis. They're identical to the original parent cell. And so if this was a skin cell, it's going to go through its cell cycle, behave like a skin cell, do all of the things a skin cell needs to do, and then will divide and produce two new daughter cells that are each identical copies of skin cells, which is the way that your skin will repair itself. Bone cells produce bone cells, nerve cells produce nerve cells, and so on. So when we focus in on an actual mitotic image of a cell in Interphase, you would see a cell that looks like this. Notice that there really isn't any kind of organization to the nucleus. It looks very uniform. That's because the DNA in this case would be in a form of DNA, which we will talk about in more detail later, but is referred to as chromatin.
And so the cell spends the majority of their time in interphase. We sort of alluded to that on the last page, that interphase is G1, S, and G2. And so if you look at our graphic that's now up in the corner there as a sort of a pie chart or a percentage, you can see that interphase takes up the vast majority of that time, and that is true for the cell, that it's going to spend most of its time in those three subphases.
And the majority of, like we already talked about, the majority of the cellular processing and cellular energetics and all of the things the cell needs to do happens in that phase.
So let's hit on some of those big important functions that are happening in interphase. The first big one there, metabolic reactions. We just got done with photosynthesis and cellular respiration as our units. These reactions and functions occur throughout interphase. This is where those cells are doing those things. We're producing ATP for the cell, we're converting sugars, or we're producing sugars depending on cell type, but those are being carried out during interphase.
The second one would be protein synthesis. We're going to cover this in a lot more detail later in a few units from now, but protein synthesis is the cell's ability to produce proteins and enzymes that are necessary for everything the cell needs to do. So that could be for the growth that occurs during G1 and G2. It could be for DNA replication, which happens in S. It could be for mitotic or cytoplasm division, and it could be for the normal cellular functioning like the metabolic reactions that we talked about before.
Yeah, last semester when we talked about our molecules, we talked about how proteins carry out pretty much all of the functions in the cell or assist with that. So that's a good list to remember, that this is what's going to be doing the jobs in the cell.
And there is a thing in biology called the central dogma that is basically fundamental to all things biology, and it has to do with DNA producing not only itself, but DNA producing RNA and RNA producing that protein. Again, all of this is coming later in the class, but we kind of can begin to talk about it now, only because we're talking about DNA duplicating itself. Organelle production, like we said in G1 and G2, a lot of the organelles are being produced. They're increasing in number and they're increasing in size, and that's because the cell is physically getting bigger. Larger cells have to include more and bigger organelles in order to support themselves, and they have to increase in number so that when the cell divides, it can actually kind of divvy up the organelles and produce two new daughter cells that have the organelles it needs.
Finally, like we talked about in S phase, we're going to replicate that DNA. This is what is going to ensure genetic continuity between cells, that we do, in fact, have the ability to replicate a cell and prove true that component of cell theory that cells come from pre-existing cells.
And genetic continuity is an important term when we talk about cells becoming identical to the cells that create them.
Exactly. So we'll dive into that more as we continue.
So when we look at a DNA molecule, DNA stands for deoxyribonucleic acid, and when we kind of go as far into the structure of DNA as we can, we notice that it is a twisted ladder. The rungs of the ladder would be nucleotide base pairs. Base pairs can be either A, T, C, or G. We're going to talk about DNA in more detail in our DNA unit, but the twisted ladder is called an alpha helix, and that alpha helix will wrap around histone proteins, which are like spools that we wrap our fishing line or thread around. Those are a way to organize the DNA and keep them from tangling and keep them more organized. Those histones will eventually coil themselves into this structure, which we refer to as chromatin.
If you are not sure what to write down the term chromatin and then the term chromosome, which is the next one there. So chromatin and chromosomes are the two forms that we will most often, besides the term DNA itself, chromatin and chromosomes are the two forms that you will see us refer to DNA as a structure within these processes. So during various phases of the cell cycle and mitosis, we will talk about DNA in certain forms. So we will mention the term chromosome or we will mention the term chromatin. So make yourself familiar with those two terms.
Yeah, and chromatin is the inactive form and chromosome is the active form. Chromatin is going to be found in the cell during interphase and chromosomes will be found during mitosis.
And so there will be a step where we sort of condense them down. As you can see in this picture of a chromosome on the right, we sort of mush the chromatin together and condense it and wrap it really tightly, and that forms those chromosomes.
Right, notice this is an X shape, but the X consists of a single fiber that is really densely packed into the structure. And so in class, we will typically draw DNA chromosomes in the form of this kind of butterfly or X shape, but it is an actual single strand of DNA that's supercoiled into that structure. There are terms we need to know. Centromere is that middle point that holds two sister chromatids together. Sister chromatids referring to a duplicated chromosome. So this would be one sister chromatid. This would be the second sister chromatid. And those two sister chromatids, while they're identical, are held together by that common protein structure, which is a centromere.
So as we go through this process, you will see a point where these things divide during mitosis, specifically in anaphase. And so after anaphase, it is important to understand that when we separate our sister chromatids, so when we actually attach what are called spindle fibers to the centromere there in the middle, and it will pull apart these chromosomes. But at that point, we do still refer to the two sides of your chromosome. The two sister chromatids are referred to still as chromosomes.
And once the sisters divide, they will duplicate themselves in their own S phase, and it will make this duplicated chromosome structure again.
So just to wrap up and highlight a few things and kind of put it all together, some big takeaways. Remember that we have most of the cell cycle is spent in interphase, where we are growing, producing proteins, and carrying out normal cell functions, with the key of duplication of DNA in S phase. Once we reach a point where our surface area to volume ratio of the cell requires a division, so we're too big, we will move into the M phase or mitotic phase, where that cell can then be divided, both the division of the nucleus in mitosis and the division of the cytoplasm through cytokinesis, resulting in our two identical daughter cells.
And they are not only identical to each other, but they are identical to the original parent. That's it for this video.
Thanks for sticking around.
See ya.